Good morning, students. Today's topic is cell lines. The primary cell cultures are finite and then enter a phase of senescence. The cells may become multinucleate, enlarge in size, lose their adhesive properties, and may lose the inherent tissue or cell characteristics. This is a disadvantage in research. Certain methods to immortalize primary cell lines are by incorporation of viral genes such as SV40 and by expression of telomerase reverse transcriptase protein. What is a cell line? Cell lines are permanently established cell cultures obtained from primary cell cultures. They could be finite or continuous cell lines, as I discussed in one of my earlier sessions. The figure below here explains the ways to obtain a finite and a continuous cell line. A finite cell line divides till the Hayflick limit is reached. Example, for human cells it is 50 to 100 times. A cell strain is a subpopulation of cell lines selected positively by cloning or other methods. It is different from a cell line. The graph here represents the growth kinetics of a continuous cell line generated from a primary cell line. Continuous cell lines are the fast-growing monolayer or suspension cultures. They are immortal or tumorigenic. A continuous cell line could be a stem cell line or a immortal transformed cell line. The stem cell lines could be derived from embryonic stem cells, adult stem cells, or induced pluripotent stem cells. The embryonic cell lines are derived from the inner cell mass of a blastocyst. For this, generally embryo leftovers for IVF procedures are used. The adult stem lines are found in juvenile or adult tissue. Example, the cells derived from hemopoietic cells of bone marrow and the mesenchymal cells from the umbilical cord and amniotic fluid, or could be adipose tissue. They are multipotent. The induced pluripotent stem cells are the ones which are derived from somatic cells. The method was discovered by Yakinaka and his team in 2006, for which he was honored the Nobel Prize in 2012. Certain terminologies used in cell line culture. Passage number. It is the number of times the culture is subcultured. Population doubling. It is the time taken by the cell population to double, and it is determined by the formula given below, where N naught is the original number of cells plated in the culture vessel, while N is the number of cells in the culture at the end of the time interval. Generation time is the number of doublings that a cell population has undergone. Note the difference between population doubling and generation number. Split ratio is the divisor of dilution ratio of a cell culture at subculturing. Example, if a flask of 100 ml is divided into 4, the split ratio would be 4. That is 1 by 4. The divisor number here is 4. Plating efficiency. The percentage of cells seeded at subculture to give rise to colonies. Seeding efficiency. The percentage of inoculum that attaches to the substrate within a stated time period. Cell concentration is the number of cells per ml of medium, while cell density is the number of cells per centimeter square of substrate. The graph here represents repetition of standard growth cycle, that is, growth kinetics curve after each serial subculture put together. Here, if you notice, subculture is done thrice. Hence, the passage is 3. It is shown in different colors. Consider each passage. The number of times doubling of cell occurs is 3. Hence, the generation number for each passage is 3. Nomenclature of cell lines. Nomenclature of several cell lines have not yet been standardized. One method is to give codes and designations to cell lines for identification. Example, NHB 2-2 means the particular cell line is derived from normal human brain cells. The cell line number is 2 and it has gone population doubling of 2. According to Society of In Vitro Biology, 4-letter acronyms can be used to designate special or specific cell lines, or initial 2 letters to indicate the institution of origin. Example, MCF7 means Michigan Cancer Foundation Breast Cancer Line 7. Other one, WI38. It means Winster Institute Lung Cell Line number 38. According to the European Human Pluripotent Stem Cell Registry, it is automated. It gives a maximum of 15 character codes and can be categorized as human embryonic stem cells with code ESC and human induced pluripotent stem cells as iPSC lines. In this method of nomenclature, first, a donor ID is created. It could be institutional name with 3-digit alphanumerical code, which starts from 001 to 999. After 999, it continues with 00A to ZZZ. The second is the clone number and third is the subclone. The first character of the code is 2 to 6 digits, which includes the name of the generator or donor. The second letter is a single digit number which denotes the cell line type, that is, whether it is embryonic or induced pluripotent stem line. The third donor ID is a 3-digit number. It could be numerical or alphanumerical. Next is the clone number, which is a 2 to 3-digit alphabetic number which proceeds after a hyphen. Then, if a subclone is there, the particular cell line, an alphanumeric number code is given after preceding a hyphen. Let's understand it with an example. Here, as an example, we'll consider the cell line UKBI001 A-1. Here, the letters UKB in blue denote the institution. The letter I gives the cell line type, that is, in this case, it is induced pluripotent stem cell. 001 in green denotes the unique donor name. The letter capital A, which precedes after the hyphen, is the donor's first cell line, and the letter 1 written in blank after the hyphen denotes the first subclone. The table here shows the list of the commonly used cell lines with its properties. The cell lines are maintained by subculturing and depends upon whether they are monolayers or suspension cultures. The methods of subculturing has been discussed in my earlier session. Each cell line should be well documented and maintained. Documentation of cell lines. Each vial or cell line should have records of the owner who last worked with it, the cell name, the donor, the date of origin, the passage number, the type, the source, the freeze date if the cells are to be freezed, the cell count if there are any special properties of the particular cell line, and the safety informations with additional comments if any. It could be recommendations for use for future experimenters. This is one of a format for documentation of cell lines or vials. It could change from lab to lab. This is an example of a documentation format if the cell lines are to be freezed or cryopreserved. Applications of cell culture technology are: they can be used as model systems for studying basic cell biology, interaction between disease-causing agents and cells, effect of drugs, processes such as aging and nutritional studies. It could be used for toxicity studies in case of study of effects of new drugs, cancer research, virology, in genetic engineering for production of commercially important proteins, large-scale production of virus for use of vaccine productions like polio, rabies, and also in gene therapy. Thank you.